Hello and welcome to Health Cities Talks. In this edition, we're going to hear from two companies based right here in Alberta that are building some really innovative health technology. PanTHERA CryoSolutions, which makes technology to support the storage of cells, and Tevosol, which makes machines for organ transplantation. My name is Mack Male and I'm the co-founder of Taproot Publishing. Every Wednesday, we publish the Health Innovation Roundup, which is a look at everything that's going on in the health innovation scene right here in Edmonton, Alberta, the latest headlines, happenings, and everything else. My job today is to shepherd us along in this conversation and find out some more information about these two really interesting companies, and I'm really excited to learn more about them. So let's dive right in. I have two guests joining me for this edition. We have Dr. Jayan Acker, who is the co-founder and CEO of PanTHERA CryoSolutions, and Dr. Jayan Nagendran, co-founder of Tevosol. Welcome to you both.
Thanks, Mac.
Thank you.
Jason, maybe we can start with you. Tell us a little bit about PanTHERA and what your organization does.
Absolutely. So PanTHERA is a company that was spun out from the University of Alberta based on technology that was developed in collaboration with Dr. Rob Ben at the University of Ottawa. We were formed in 2019 after about a decade of research on the development of small molecules that help control ice in very complex systems. Now, ice is really a critical detriment when we try to freeze cells, tissues, and organs for transplant. So one of the challenges that our technology had to overcome was how to mitigate that damage. So working with Rob Ben, who was inspired by natural systems that survive freezing, him and his team in Ottawa developed small molecules, synthetic organic compounds that are really very potent at controlling ice. So the analogy we give is with ice cream. So if you are familiar with ice cream and you leave it in the freezer too long, you get ice recrystallization occurring. That's the small ice crystals that grow into that crunchy ice cream that no one wants. Similar things happen in cell systems. So our technology is based on the ability to control that process, keep the ice crystals small, and as a result, protect those cells, tissues, and organs from damage. And we've been taking this technology now, both in the cell and gene therapy area, but working with Tevosol and Jayan, really looking at overcoming some of the challenges in the tissue and organ space.
I think during the COVID-19 pandemic, we've heard about vaccines and how some of them, like the Pfizer-BioNTech vaccine, need to be kept extremely cold to preserve them a little bit longer. Your technology can help address that challenge as well, right? That's what you're talking about with the ice crystals. Can you walk us through that?
Yeah, absolutely. So one of the challenges when you're storing a biologic, whether it's something like a vaccine or cell-based therapy, is that getting the cells down to that temperature where it is stably stored and then getting it back, you have to transverse temperature extremes. And it's during that movement from a low temperature to a high temperature that those ice crystals that do form in the system can actually grow from very small crystals to very large crystals. And as a result, damage the cell therapy or the biologic that you're trying to preserve. So when you're transporting a vaccine or transporting a cell therapy product, you need to store it at extremely low temperatures to prevent that from occurring. Now, our technology actually does two things. One is it keeps those ice crystals small in the first place, so you don't have the damage from that. But also during that transient warming that can occur during transport, it protects those biologics so you can either have better outcome at the end, or in the case of our technology, you can actually store them at higher sub-zero temperatures. So you don't need to be shipping on liquid nitrogen, for example, or at ultra low temperatures. So it gives you better stability of that product and get that product to the end user and allow it to be effective.
Okay, great. Well, Jayan, let's bring you in here. Why don't you tell us a little bit about Tevosol?
Yeah, thank you for the opportunity, and I appreciate the chance to have this conversation with you and Jason, who's a great collaborator and colleague. So my background is I'm a cardiac surgeon. I'm also a cardiothoracic transplant surgeon, and my interest is in organ preservation. And my colleague, Dr. Darren Freed, who's also a cardiothoracic transplant surgeon, actually a pediatric heart surgeon. He and I had started the first in Western Canada ex vivo organ perfusion center. And what that is we take organs that are potentially used for transplantation out of a donor and put them in an environment where they could be kept alive and safe longer. The reason being is that currently, out of the few donors we have, we turn down more than 75% of the lungs that are offered. So of those few precious potential gifts of life, very few of them are accepted. in part, that's due to reversible causes within the donor that we're unable to change as they're in the donor. And oftentimes we just say no to a set of lungs that the day before, whatever went on to make that person a donor happen, those were good organs. So by liberating them from the body and putting them almost in a robot chest, we're able to show many organs that we would say no to are actually usable for transplantation. So after working in the lab for a few years together from 2013 to 2015, we incorporated into Tevosol to then, out of the University of Alberta, to then try and actually use this technology clinically. And we've been successful with that with our lung technology. We developed a normal way for lungs to breathe outside of the body called negative pressure ventilation, and kept those lungs alive with a blood-based solution in them. And we took 12 sets of organs that were turned down for transplantation and would have otherwise been discarded, and converted all 12 of them into usable organs and transplanted 12 patients that otherwise may not have received a transplant. So with those successes, we've gone on to look at how our platforms could be used in collaboration for other technologies to be introduced onto our platforms to maybe extend preservation. And certainly there's potential for synergy with what PanTHERA is doing in looking at potentially taking organs into this cryopreserved state so that they can actually be banked and we can decide when it's time to do a transplant and then bring them back to an animated state where they can be evaluated on a technology like ours, potentially for future transplantation. So we're right now in the process of doing a pivotal trial with the FDA to commercialize the lung technology, which is called the EVOS, which is the ex vivo organ support system for the lung. And we hope to follow that up with the heart in future years. And we've been fortunate to have been acquired by a company, Bridge to Life, that's helping with our commercialization efforts.
So not only improving the quality of transplants that happen, but the quantity as well, because you're saying more organs are now available for transplantation. That's really interesting. And you mentioned the heart there quickly. I know you want to have this solution applicable to a variety of organs. Was there any reason you started with the lungs in particular?
That's a great question. And indeed, I am a heart surgeon. And the reason why, and I also do lung transplants, but the reason why we started with the lungs is because there were developing technologies in the fields attempting to do this, to keep lungs alive outside of the body by ex vivo perfusion. What we realized is that they lacked significant penetration or adoptability for a variety of reasons, many of them due to the efficacy of the technologies available to date. We felt that if we as surgeons who are also iterating things in the lab brought that expertise to the design component, we could really understand what the organ needs, working with them clinically every day, and also what the surgeon needs to allow for this technology to be used readily, reproducibly, and actually add to the potential number of transplants being performed. And so it was with that mindset that we created a device that truly houses the lungs in a natural way. No other device breathes for the lungs. Other devices might put a breathing tube down the lungs like you do in the intensive care unit and blow air into those lungs. Whereas our device, just like your chest and my chest, the device is sealed in a container and a vacuum is applied, just like when we breathe and our diaphragms contract, and air naturally goes into the lungs by negative pressure ventilation. And simply by adhering to some of these basic principles of physiology, we were greatly able to improve the quality of these organs. And so that's why we started with the lungs, and also the most amount of donors that are, patients that are dying waiting for transplant is also on our lung list, where 1 in 3 patients die waiting for lung transplant. So it's pretty tragic that we turn down over 75% of the offers we get.
Absolutely. I think it's so interesting when we think about health innovation and applying technology to the body. We often think about putting technology in the body. I think that's the popular culture idea. And so it's fascinating that you've gone the other way, taking the organs and helping them survive in a better quality state outside the body. You've both mentioned that there's some opportunity for you to work together. Jason, when I hear cryogenics, I think when most people hear cryogenics, they think rich people trying to preserve their life or make it to some distant point in the future. what are some of the applications of the technology that you're working on, and how does that apply to what Jayan's told us about what Tevosol's doing?
Great question. Cryopreservation is a technology that really is an enabling technology for the future of what medicine is going to look like. We're moving from a time now where we're developing drugs and pharmaceuticals to developing cells and cell-based therapies to reverse and repair and treat a number of illnesses and diseases. Many people will be familiar with stem cell transplants or the donation of cord blood and the stem cells that are in those to help treat cancer patients and a variety of other disease. And that was really one of the first early cell therapies that we were able to roll out. And many of the groundbreaking discoveries on the preservation of those cells occurred here in Edmonton in the late 1970s and early 1980s. So we've taken that same strategy as we're developing these new cell therapies, whether it's a new therapy to treat cancer, whether it's a new therapy to treat diabetes, whether it's a new therapy to treat immune disorders. All of them are going to involve cells, and stabilizing those cells is very important. And our technology not only allows you to improve the quality of those cells, but allow you to allow the manufacturers of those cell therapies to do it in a way that's more efficacious and easier to deliver that technology to the end user. So that's the immediate market. But this technology equally has applications, as we're discussing with Jayan, where ice is an incredibly damaging thing that occurs when we try to cryopreserve tissues or organs. And to date, there's been very little success in cryopreserving tissues and organs because of this challenge with ice. So we're taking an approach of using these ice control agents, these ice recrystallization inhibitors, to try to minimize that. Now, with the cells, it's relatively easy to get these small molecules in. But when you start to scale up to the size of a tissue in the millimeter scale, or the organ now in the centimeter scale, you really are challenged by the size of the organ. So how do you get these cryoprotectants, these ice recrystallization inhibitors inside? And that's where Tevosol technology really shines. And it gives us the ability to disperse these ice recrystallization inhibitors in a very uniform way outside of the body, obviously, as part of that preservation strategy. And then the Tevosol technology has the further advantage is it allows us to actually evaluate the quality. So we can actually store the organ in its more natural state for a period of time while we're evaluating the toxicity of these cryoprotectants, but also their efficacy, and getting the organ ready to then cool it down to sub-zero temperatures. So the work that Jayan and I are working on, both in the liver and in lung systems, are really geared towards storing now these organs in a super cooled state. So below zero, where ice now becomes a problem, we have to mitigate it. And that's where the PanTHERA technology really shines. But equally, this technology has applications in reproductive medicine. We're doing work with collaborators at the Smithsonian, looking at using these for conservation biology applications. Aquaculture, we have funding working with groups that are interested in using these to cryopreserve fish embryos, of all things, to help the aquaculture industry. So, cryopreservation is a broad technology and has huge applications.
Very interesting. Okay, so what the two of you are doing, the two companies are working on, sounds really innovative, obviously, but also kind of unique in that you're able to kind of collaborate in this way that might not be possible in other jurisdictions or in other places. What is it about Edmonton or Alberta that makes it possible for the two companies to come together? And maybe, Jayan, we can start with you and your thoughts on that.
Sure. So, having trained at the University of Alberta, having been here since an undergraduate student, and then my medical degree, and then my training in cardiac surgery, and then my PhD, I did go to Stanford University to do heart and lung transplantation. And I was exposed to a different environment, though a highly academic, highly productive environment, different means by which collaboration and synergy could be approached between specialties. And it's very defined and somewhat challenging. Whereas at the University of Alberta, it's clear that there's a environment to foster these collaborations, and there's very few barriers to do so. As a result, there's also the encouragement from faculty support onward to look at translational medicine technologies, things that can actually be developed in the lab, like myself and like Jason's lab, that can be translated into human applications with actual clinical results being evaluated here locally as well. Because of our robust medical program and hospital system here in Edmonton, that is directly attached by physical building to the laboratories that we work in, we're able to do this somewhat seamlessly. And one of the examples is that, Jason and myself now have co-supervised multiple graduate students. And as a result, they get the exposure to the things that are on the surgical side that might help with understanding how some of the technologies, Jason has developed could be applied. And vice versa, they bring to my lab the understanding of what it takes to allow for these types of technologies to permeate tissue and evaluate that permeation and evaluate the survivability of those tissue slices or cell cultures to better understand how we could maybe deliver our technology to be more safe in creating something like a virtual organ bank. Right now, we can keep organs alive for maybe a day or two, but with Jason's technology, we could potentially have a bank of organs that when it's a perfect match for somebody, that's when we bring them out of that state of cryopreservation for transplantation. So I would say the fostering of a collaborative environment is much more free-flowing at an institute like the University of Alberta, and highly encouraged, and it's appreciated.
And Jason, anything from your perspective to add to that?
Yeah, no, Jayan, I really summarized the ecosystem here really well, where we're able to work seamlessly, and it turns into a whole bunch of other interesting projects. And my work with Canadian Blood Services, for example, there's a natural discussion now with Jayan and Darren around blood products that are used in these ex vivo. So it just continually grows on itself. But just probably wanted to add that the University of Alberta is a world-class center of excellence for cryobiology and cryopreservation, and it has been for over 50 years now. So we are a great group to be able to sort of challenge some of these problems that the organ preservation teams are having around stabilization and stability, and take the technology that is being developed in the basic research labs right through to translation and clinical translation. And I think we're unique to do that. Edmonton is not, it's not surprising perhaps that we have a world-class cryobiology research group here. We have winter 9 months of the year, so you got to do something when it's cold.
I might quickly add to that, and Jason's absolutely right, not just on the cryopreservation side, but if you look at the evolution of cardiothoracic transplantation, the University of Alberta has the second largest cardiothoracic transplant program in Canada, next to Toronto. And in the rankings of lung transplant programs in the world by the International Society of Heart and Lung Transplantation, we're in the top 5% of the world for volumes done per center, doing over 75 double lung transplants a year. So we have a very large, very robust clinical program to then draw from and take back to the lab in these translational efforts. So we're fortunate that way, that both are strong and both are accessible immediately.
Don't you just love it when the cold of Edmonton and Alberta turns into a positive, and some incredible things come out of that. You've both had quite the pandemic in terms of progressing the companies. I know PanTHERA raised some money in kind of 2020, and Jayan, you mentioned the acquisition. Maybe tell us a little bit about how getting that money from outside, bringing that to Alberta. What is the kind of impact that has not only for your organizations, but for the ecosystem here? And Jayan, maybe we can start with you. I know you had quite a bit of success getting Tevosol started from Albertans, and now you've gone to that next level. Tell us a bit about that.
Sure. I feel very humble. There's tremendous grassroots support in Alberta to swing for the fences, to look for innovative, different ideas. And that support is deep. There's generous pockets, and we've been able to raise close to $5 million truly from friends and family and word of mouth by developing this. And that was leveraged into performing a first-in-human clinical trial where we took those 12 sets of lungs that wouldn't have been used. And that's unheard of, unprecedented for such a small amount of investment to lead to a medical device that was used in humans within a few years. And that says a lot about what we can do here in Alberta. And that had led to the recognition internationally, where a company like Bridge to Life, who was looking to acquire perfusion-based technologies for transplantation, really came to us to see what we were doing and realize that we had something that could be the real deal, so to speak, and commercialized. And that wouldn't have been possible if it wasn't from the initial investment from Albertans. And hopefully those investments will continue to grow for those who invested. But on top of that, it's also led to novel creation of jobs in the medical technology device space, not just with manufacturing, but with hardware engineering, software engineering, and also even in the development of novel solutions and preservation solutions for our technology. So it's been a pleiotropic kind of process that early initial investment, along with support from the Canadian government through the IRAP program, has allowed for us to really do more than we ever could in more limited jurisdictions.
And Jason, you've had a similar experience, I think, and also been able to benefit from some of those government programs.
Yeah, absolutely. The technology would never be developed if it wasn't for the investment from the research organizations like GlycoNet and Canadian Blood Services and CIHR and NSERC, and then having the ability to translate that into the government programs like IRAP that Jayan mentioned. But ultimately, you do need to raise the money to get it to the next level, and we were very successful in identifying BioLife Solutions out of Seattle, which is the world's leading provider of biopreservation solutions. They saw the opportunity and early on made a commitment in investing not only in the company, but also the further co-development of this technology. So it's been great. All that money is coming into Alberta and into Canada, and we're able to use it to hire in a variety of areas, not just in the science, but also now in everything from marketing and quality systems and manufacturing. And it's been great. I think it's been a great boon for us to try to get this investment here in Alberta and create jobs that are going to help then spur the next round of the ecosystem development. So we need successes like Tevosol and PanTHERA.
Absolutely, yeah. And building on those successes is the secret sauce we're trying to figure out. Where do you think PanTHERA could go? What is your hope for the organization and the technology that you've built? Like big picture, what do you think is the real opportunity?
Well, cells and tissues is cracking the surface. We're gonna be able to provide a product for end users of cell therapies that are really going to help them deliver on the clinical and therapeutic outcomes that they're looking for, and make it more cost effective and efficient for them. And that's just going to then trickle into some of the other areas that I've discussed. The work that we're doing with Tevosol. I'm extremely a pathologically optimistic person, and I love to see. Be in that operating room when Jayan's transplanting that first organ that we've thawed, and seeing the success from it. And I think we have the opportunity to do it here at Edmonton. So again, stay tuned, and hopefully that's what the future is going to look like for us.
Be a bit optimistic to be an entrepreneur, so I get that for sure. That's fantastic. Jayan, a couple of years ago, you pitched at the NASA iTech competition and won, actually. And I think what you were talking about was potentially using your technology to support astronauts in sort of future long-distance space travel. That sounds like a big future potential opportunity. Same question to you. Where do you see Tevosol and the work you're doing going, and what is the big opportunity that you are envisioning?
Sure. So I'll start by talking a little bit about the NASA competition, and then about the future as well. And so, yeah, in the NASA iTech competition, we did end up winning as a startup company with some of the potentials for space application. In that right now, there are big projects looking to send astronauts to Mars and even set up colonies there. And a large part of that involves potentially putting astronauts into a state of torpor, or beyond deep sleep, or hibernation, if you will, to allow them to get there. And instead of testing some of those cocktails and technologies on actual humans, with our ex vivo organs, you can actually test the toxicity and efficacies of those potential treatments to induce those states outside of the body, simply on organs itself, to first identify if this might be safe, to then trial on potential astronauts in the future, as well as the pharmacology of how some medications work in space. Before trialing it on patients, you can actually take organs potentially into space and trial it on organs as well, to understand what the physiologic effects are. as well as exposure to radiation. In the cosmos, there's significant exposure to radiation that these astronauts are exposed to, and with our technologies here on Earth, we can examine what some of the mitigating strategies might be that are effective in minimizing the risk of all of that exposure. And those were some of the ways that our technology may be applied in deep space considerations. But further, and here back on the terrestrial ground of Earth, we see that there's an opportunity to improve the quality and quantity of organs being transplanted, which it doesn't matter how those organs are developed. You saw that there was a first in human pig heart transplant performed by Dr. Bartley Griffiths at the University of Maryland a month ago. And though that's still in its infancy, that xenotransplantation is a big step. But before that's going to become prime time, surgeons and patients are going to want to know that those organs are functioning well. And that's where our technologies and platforms come in, that we can actually, if it came from a modified pig, or if it was grown in a lab on a bioscaffold that was then populated with the right cell types to create the organ outside of the body, we still have an opportunity to evaluate it before deciding if it's appropriate for clinical transplantation. And then when you look at the bright future and collaborating with people like Jason and PanTHERA, if we do develop that cryopreserved organ bank, that are first bringing those organs into that cryo state by using our technologies to allow for the introduction of his technologies to allow them to be cryopreserved, and then reanimating them at the time of surgery with our technologies, then you can see that many diseases right now that we don't have a good cure for could potentially be transplanted. We also may be able to have the best immunologic match by treating these organs outside of the body to change their cell surface antigens so that they're more appropriate for transplantation without rejection. So the future is really bright. I think it's happening in many ways faster than we think. And here in Alberta, I think we're really at the crossroads where we can be international leaders and a center of excellence. We are a center of excellence in the types of research we do. I think we can be centers of excellence in the technologies we bring to the world, and as a result, really diversify our portfolio as an economy to be a manufacturing and production expert of these technologies too. You think about the diversification from traditional oil and gas. Well, some of the machine work and things that are done in NISCU are absolutely applicable to the development of the machines and technologies we're building to do outside of the body as well. So I do see great opportunity.
Absolutely, and there's some really incredible things happening in Alberta, your two companies, of course, but a variety of other things as well. Jason, what advice or guidance would you give to other entrepreneurs in the space who are looking to have such a positive impact on the future of human health, like PanTHERA and Tevosol?
Well, it's maybe not surprising, but be bold. I think they sometimes at the University of Alberta we don't realize the amazing wealth of resource, both in equipment and access to things like what Jayan's talking about in terms of the clinical side. But we also have amazing people, and I think one of the challenges that we have to do is really encourage those those individuals to be bold on the academic side and take that great idea and really see what kind of legs it has on it, and can you actually get it out of the lab and into the clinic or into the industry. And that's going to require, again, training. We need to do a lot more on the training and education of entrepreneurs, giving them the opportunity to work with successful entrepreneurs and having those role models so that they can see. And then, for Jayan and myself, making sure that there is those opportunities for collisions to occur between the basic researchers and the clinicians, so that when, Jayan and I are sitting and he's expressing a problem he's having, and I see a solution, and similarly, I have a solution or he has solutions for problems that I hadn't even anticipated. So, it's that collaborative environment that you have to create. And I think universities are a phenomenal place for that to occur, and particularly here at the University of Alberta.
Well, we've reached that time of the conversation. This has been fascinating and very informative. Thank you. I want to give you both an opportunity to leave listeners and viewers with any sort of last closing remarks, or maybe even a call to action. Is there something that the folks out there who might be watching this could do to help take your organizations and your initiatives to the next level? So Jayan, you want to start?
I think that the city of Edmonton is again well poised to be a leader in developing international medical technologies, and to that level, I would continue to support the city and the province to be bold and fund projects like Tevosol and PanTHERA, because those initial investments have the potential of being so much more, not just when it comes back to the ROI of that investment, but what it does to enrich the society and the type of people that we're hiring now, bringing into Edmonton, who are highly qualified professionals, enriches the province, enriches the city. So I think my take-home message is, and to the people who want to start, it's not an easy journey. It certainly takes a lot of tenacity and perseverance, but there are supports here. This is a great environment to be in to try and start. And if you look for mentorship, mentorship is available. And that also is a great strength of being in this city. I've certainly had many great business mentors now, as I had medical mentors when I was in training. Now on the other side of the coin, help me along this process and journey as well. So I think those are things to really appreciate here in Edmonton and in Alberta.
Absolutely, some incredible mentorship programs and opportunities at the U of A and elsewhere in the city. Jason, any closing thoughts from you or call to action you'd like to share with viewers?
No, again, for the young person who's listening to this and wondering what opportunities there are in Edmonton, be proud of the fact that we are developing this ecosystem, that there are going to be opportunities for you when you leave high school or when you're leaving your university to stay in the province and really look for those opportunities, whether, I hate to say it, whether it's in Calgary or whether it's here in Edmonton. There are opportunities and there's great things happening. Explore them, reach out to individuals that can provide that mentorship. But stay here, stay in Alberta. There's great things happening. And I think with the success of Tevosol and PanTHERA and many other companies that are doing extremely wonderful things here in the province, we'll hopefully have those opportunities for those very talented people to stay and make Edmonton their permanent home. And I'd love to see that. I think we have a lot of phenomenal international students that come to Alberta looking for that opportunity. I think finally we have now something for them to stay in and make Alberta and Canada their home.
Absolutely. Okay. Well, fantastic. Thank you so much, Dr. Jayan Nagendran from Tevosol and Dr. Jason Acker from PanTHERA CryoSolutions. It's been really fantastic talking with both of you. I know you're both very busy and doing some incredible work, and I really appreciate your time. And thank you to everyone else who is out there listening and watching this. You can find previous as well as upcoming episodes of Health Cities Talks at healthcities.ca, as well as all of the other really great work that Health Cities is doing to help support and grow the ecosystem here in Edmonton and Alberta, and indeed across Canada. And also be sure to check out the Health Innovation Roundup from Taproot Edmonton at taprootedmonton.ca. Thank you.
Awesome. Thanks, Mac.